Imagine trying to build a human brain. Like, you aren't just slapping parts together on an assembly line.
Right. Yeah, it's not that simple.
No, you're starting with just a single microscopic stem cell. And here is the truly wild part. Every single cell in your body, from the neurons in your cortex to the skin cells on your toes, they have the exact same DNA.
It's the exact same master instruction manual.
Exactly. So how does a single cell actually know whether it is supposed to, stay a generic dividing stem cell, or if it's supposed to transform into this highly complex, specialized neuron, like the kind that helps you process a childhood memory or solve a math problem?
It really is the ultimate biological paradox. Because the genetic blueprint is completely identical everywhere, the secret has to lie entirely in how that blueprint is read.
Right.
For decades, the assumption in biology was, well, relatively straightforward. We thought we just needed to know which genes were turned on or off, like a simple binary code.
Just a bunch of zeros and ones.
Exactly. But as we look closer, we realize that the process of acquiring a neural identity is actually an incredibly intricate, multi-layered physical mechanism.
And that is exactly what we are unpacking today. Welcome to our deep dive. We are looking at a massive stack of research, but the absolute crown jewel is this groundbreaking paper published in Nature Neuroscience by Noack and colleagues in 2022.
It's a phenomenal piece of work.
It really is. They essentially built a molecular roadmap of the developing brain, and we are going to explore the 3 hidden layers of gene regulation they uncovered. We've got the workers reading the blueprints, we've got the hidden switches scattered across the genome, and finally, the actual 3D folding of the DNA itself.
Yeah, and to understand the sheer scale of what this team accomplished, we have to look at where they started. The researchers zeroed in on embryonic day 14.5 in the developing mouse cortex.
Which is a very specific time frame.
Very specific. And that's because it's mid-neurogenesis. It's the absolute peak rush hour of brain development. Millions of cells are making their life-altering career choices all at once. Yeah. And to capture what is happening, they used a technique called single-cell RNA sequencing, or cRNA-seq.
Okay, let's unpack that, because single-cell RNA sequencing sounds, well, intensely complicated.
It is a bit of a mouthful.
But if we use a factory analogy, it's kind of like freezing time in the middle of a chaotic, busy shift, right?
Yeah.
And taking a high-resolution snapshot of exactly which blueprints the RNA molecules are being actively photocopied by thousands of individual workers at that exact millisecond.
That is a perfect visualization. You are capturing the immediate thoughts of every single cell. And when they took that snapshot, they could actually trace the lineage journey of these cells.
Like watching the cellular pipeline in action.
Exactly. It starts with neural stem cells. Those are the foundation. Then those stem cells transition into what we call intermediate progenitor cells, or IPCs.
Okay, so middle stage.
Right. And finally, they mature into projection neurons. Those are the specialized cells that will actually send electrical signals across the brain.
What I found so fascinating in the notes is that this journey, it isn't a smooth, gradual ramp-up. It happens in these very distinct overlapping transcriptional waves, like a relay race.
Yes, the timing is incredibly precise.
The researchers observed that in the foundational neural stem cells, a specific gene called HES1 is highly active. But as the cell decides to become an intermediate progenitor, HES1 drops off, and a new wave surges forward. Specifically, master regulators with names like neurog2 and EOMs.
And then, as it makes that final leap to become a mature projection neuron, a third wave crashes in. That one features genes like neurod2 and MAPT. The baton is passed perfectly from one gene network to the next.
It's a beautiful choreography.
It is. And we have to emphasize that this isn't just abstract theoretical biology. The researchers mapped how these specific developmental waves are deeply tied to severe clinical conditions in humans.
Right, because timing is everything. For example, they tracked a gene called FHL1, which is involved in muscular dystrophy, and THD7, which is a chromatin remodeler linked to CHARGE syndrome that's a severe developmental disorder.
They even tracked a gene called LRF5.
Yeah, the cell adhesion protein. That one really caught my eye. When something goes wrong with LRFN5, it's heavily linked to autism and intellectual disabilities.
It makes perfect sense when you think about the mechanism. If a developing neuron misses its precise timing window to express cell adhesion proteins like LRFN5, it literally cannot connect to its neighboring neurons correctly.
The physical wiring is just altered.
Exactly, all because a gene fired a few hours too late. So seeing exactly when and where these genes activate gives us a massive clue about the mechanical origins of these conditions.
Wait, hang on. I'm trying to picture this, and I'm a bit hung up on the snapshot analogy.
Okay, sure.
If the researchers took a single snapshot in time, literally pulling all these cells from the exact same physical mouse brain at the exact same millisecond, how on earth do they know the order of their development?
That's the big question.
Because that's like looking at a still photograph of a crowded train station and trying to guess who is about to walk onto a train and who just got off.
That is a brilliant point, and it brings us to a highly sophisticated analytical algorithm they used called RNA velocity and pseudotime.
Pseudotime. Okay, how does that work?
Well, to understand this, you need to know how RNA is actually made. When a gene is first copied from the DNA into an RNA strand, that fresh RNA includes extra non-coding pieces called introns.
Okay, so it's got extra junk in it.
Exactly. We call this unspliced RNA. Over time, the cell edits that RNA, cutting out the introns and pasting the final instructions together to make a mature, spliced RNA molecule.
So it's the difference between a rough draft with all your messy, crossed-out notes versus a final, edited manuscript.
Precisely. By looking at the ratio of rough drafts to final manuscripts inside a single cell, the algorithms can predict the trajectory of that cell.
Oh, wow.
Yeah, so if a cell has a massive amount of unspliced rough draft RNA for the NROD2 gene, we know it's currently ramping up that gene's activity. It gives us a mathematical arrow pointing to exactly where that cell is heading in the immediate future.
That is basically molecular time travel.
It really is. That's how we reconstruct the entire timeline or pseudotime from a single frozen photograph.
Okay, let's do a quick mini recap of our first subtopic here, just to lock it in.
Sound good?
So, bullet point 1, we used single-cell RNA sequencing to freeze time and look at the blueprints of thousands of cells. Bullet point 2, we used RNA velocity looking at rough drafts versus final drafts to map the exact pseudotime of a cell's birth. And bullet 3, we saw that specific waves of genes turn on and off, and if that timing breaks, it leads to severe conditions like autism or muscular dystrophy.
Perfect summary.
So, question for you listening right now. If you could take a snapshot of the transcripts, like the dominant thoughts running through your own brain during a major life transition, what would they look like?
You'd probably see distinct waves of changing priorities, right?
Mm-hmm.
Like panic, then planning, then settling in.
Exactly. The developing brain is doing the exact same thing on a molecular level.
And that transition from planning to execution is a great way to think about it.
Yeah.
Because knowing what genes are active is really only half the battle. We need to know how they are turned on in the first place. Who is flipping the switch? Which brings us to the second layer of this research chromatin accessibility.
Okay, so to figure out the how, the researchers ran a completely different technique in parallel called scat-tac-seq.
Yeah, let's visualize what chromatin accessibility actually means. Imagine that the entire genome inside the cell nucleus is a massive, highly secure, deeply hidden library vault.
Okay, I'm picturing a giant underground vault.
Right. The DNA is spooled incredibly tight around structural proteins. This effectively locks the books of the genes away so they cannot be read by the factory workers. The promoters of a gene are like the heavy steel front doors to these specific vaults.
So if the door is locked, the gene is off.
Exactly. But what the researchers found is that the heavy steel doors aren't the whole story.
The real magic happens at enhancers, right? The paper refers to them as distal cis-regulatory elements.
Yes. If the promoter is the vault door, the enhancer is a hidden master switch located 3 floors up in a completely different wing of the library.
Oh, wow. So you flip that remote switch, and it miraculously unlocks the vault door downstairs.
That's it. And by integrating their RNA data with this new ATAC data, the team discovered over 16,000 positively correlated pairs between these distant enhancers and their target genes.
16,000 pairs. That is a massive control board.
It fundamentally changes our view of how development works. They found that the promoters, those vault doors, actually stay mostly static. They're generally accessible throughout development.
Wait, really? So the doors are just open?
Yeah, mostly. It is the distant enhancers that are highly dynamic. They are the true master control panels.
Here's where it gets incredibly fascinating. The researchers tracked when these master switches flip open. You would naturally assume a switch flips, and the light instantly turns on, right?
That would be the logical assumption.
And for some stem cell regulators, like SOX2 or PAC6, that is exactly what happens. But for other genes, something entirely different occurs, a mechanism called lineage priming.
Lineage priming is critical to how complex organisms build themselves. The researchers found that certain master switches, specifically those bound by our star player, the pioneer transcription factor, NROG2, actually get forced open long before the target gene is ever expressed.
So it's early.
Very early. NROG2 acts as a pioneer. It dies into the tightly packed, locked chromatin, finds its specific binding motif, and forcefully pries the chromatin open. It prepares the genetic landscape for a cellular transformation that hasn't even happened yet.
It is setting up the scaffolding on the construction site weeks before the concrete is actually poured.
Exactly.
But the notes mention that neurog2 can't always do this alone. It needs the right conditions. The data showed it often needs cofactors, helper proteins like MYZ1, or it needs the absence of repressor proteins like HES1.
Right, because if HES1 is physically sitting on the switch, neurog2 can't flip it.
Ah, so it's a competition.
It is. Which tells us that a cell's destiny isn't determined by a single loud signal. It's an intricate, highly sensitive calculus of activators, cofactors, and repressors, all fighting for access to these distant, hidden switches.
Okay, let's do our second mini recap here.
Go for it.
Bullet 1. The genome is a locked library, and while promoters are mostly static doors, the distant enhancers are the dynamic switches. Bullet 2. Pioneer factors like neurog2 engage in lineage priming, prying open these switches long before the cell actually changes identity. And bullet 3. This only works if the delicate balance of helper proteins and blocking proteins is just right.
Spot on.
So, listener, have you ever noticed how setting up your workspace, getting your coffee, turning on specific music completely dictates your actual work output later on?
You're essentially doing what neurog2 does for a developing stem cell. You're priming the environment.
Exactly. But as any scientist will tell you, observing an open switch in a genetic library is just circumstantial evidence. Correlation is not causation.
Right. To truly understand the mechanics, you have to definitively prove that this specific switch actually controls the lights.
Which brings us to the next massive hurdle the team cleared. Proving these switches actually work in the living brain. And they didn't just test this in some generic petri dish.
No, they used a technique with a deeply sci-fi name.
The massively parallel reporter assay, or MPRA in vivo.
The mechanical elegance of an MPRA is staggering. The researchers took thousands of these specific enhancer sequences that they had just identified computationally.
They physically synthesized them in the lab, right?
Yes, and they attached a unique genetic barcode to each one. Think of the barcode like a tiny readable luggage tag. Then they put these tagged sequences into a reporter plasmid.
And a plasmid is just a circular piece of delivery DNA, right?
Exactly. It's designed to produce a detectable signal if the enhancer gets activated.
But getting that delivery DNA into a living, developing embryo, that blew my mind. I was reading the methodology, and they electroporated these plasmids directly into the brains of living mouse embryos.
Electroporation is a brilliant technique.
Walk us through exactly how that works physically, because it sounds intense.
It is intense. You essentially apply a very brief, highly controlled electrical pulse to the tissue. That electrical field temporarily destabilizes the cell membranes, creating microscopic pores.
Like blowing tiny holes in the cell wall.
Temporarily, yes. And during that split second when the pores are open, the barcoded plasmids slip inside the brain cells. Then the pores close, the cells heal, and development continues.
That is wild. They literally shocked these custom genetic switches into the developing brain.
They did. So after waiting a few days for the cells to develop into mature neurons, they extracted them and sequenced the barcodes to see which luggage tags had actually reached their destination.
Basically checking to see which switches had turned on.
Exactly. And the results were definitive. The activity of these enhancers in the living brain was highly cell-type specific.
Meaning an enhancer they predicted would only work in an intermediate progenitor cell, actually only fired in that specific cell.
Right. It stayed completely silent in the stem cell. This perfectly validated all of their computational predictions.
But they didn't stop there. They wanted to prove exactly why the switch works.
Yes, the mechanism.
They hypothesized that transcription factors, those specific worker proteins like neurog2, were the actual physical keys turning these enhancer switches. So they intentionally mutated the DNA sequence.
This is the crux of the experiment.
They went in and changed the exact chemical binding motif where neurog2 is supposed to dock.
And when they mutated that specific transcription factor motif, it completely abolished the enhancer's activity.
The switch was dead.
Completely dead. This proves beyond a shadow of a doubt that it isn't just the general neighborhood of DNA that matters. It is the precise physical binding of the neurog2 key into that exact DNA lock that drives the development.
It's the ultimate proof of causation. Let's do our third mini recap. Bullet 1. We used massively parallel reporter assays, or MPRA, to test thousands of enhancers at once. Bullet 2. We used electroporation to shock these sequences into living embryonic brains. And bullet 3. By mutating the DNA, we proved that specific keys like neurog2 are strictly required to unlock these genetic doors. If you break the lock, the whole system fails.
A textbook validation.
So question for you, what's the neurog2 in your own daily routine? Like what is that one hyper-specific trigger, maybe a specific alarm sound or a certain colleague's approval that, if removed, just stops your whole productive process?
It's fascinating to think about finding that specific trigger, but understanding that trigger biologically actually opens up the final and perhaps most mind-bending question of this entire study.
Oh yeah, the physics problem.
Exactly. We know the RNA blueprints exist, we know these remote enhancer switches work, and we've proven it in living tissue. But we still have a massive physics problem. How does an enhancer switch that is located thousands, sometimes hundreds of thousands of base pairs away from a target gene physically reach the vault door to turn it on?
And this is where we have to completely change how we visualize DNA. We have to talk about the 3D genome and a technique called immunomethylhike.
Which is quite a mouthful.
Yeah, but we are basically talking about the 3D shape and folding of the DNA, right?
That's right. When you hear high C in this context, don't think of the fruit juice box. Think of high contact.
High contact, I like that.
Imagine the genome is a long, incredibly stiff electrical wire. If the master switch is way down at one end of the wire and the vault door is way down at the other end, the only way they can mechanically interact is if you physically grab the wire and bend it until those two distant points touch.
You have to loop the DNA. The 3D architecture of the genome isn't a straight line at all. It is constantly folding, twisting, and looping to bring these distant switches right up to the front doors of the genes.
Precisely.
But wait, if the wire is just bending and folding everywhere inside this tiny nucleus, wouldn't switches constantly be bumping into the wrong doors and turning on the wrong genes?
That would be absolute chaos. And it's exactly what the cell must avoid to survive. To prevent this, the 3D genome is strictly organized into what we call topologically associating domains, or TADs.
TADs. Okay, how do those work?
Think of TADs as heavily insulated rooms within the nucleus. The cell builds physical walls. During differentiation, as a stem cell transforms into a neuron, the entire 3D genome radically reorganizes itself.
Oh, so it's shifting the walls around.
Yes, it reinforces the insulation at these TAD boundaries to ensure that a highly active switch in the living room cannot physically reach through the wall and accidentally flip a light on in the kitchen.
So it's building walls and isolating the circuits.
Yeah.
But who is the electrician? Like, who is actually doing the rewiring, bending the wire, and building the loops?
The researchers dug into this, and it brings back our star player, neurog2.
Neurog2 again.
It turns out neurog2 isn't just flipping switches. It is fundamentally remodeling the 3D architecture of the cell.
And the mechanism for how one single protein does all of that is fascinating. The paper outlines a very clear multi-step process. First, neurog2 targets a tightly locked piece of DNA and mediates what is called DNA demethylation.
Let's break down demethylation, because methyl groups are essentially chemical padlocks, right?
Exactly. Methyl groups are bulky chemical tags that attach directly to the DNA. They physically block the cellular machinery from reading the genes, and they attract other proteins that spool the DNA incredibly tight.
So neurog2 orchestrates the removal of those bulky methyl tags. It strips the locks away.
It picks the lock, removing the physical blockage.
Yes. And once the methyl groups are gone, the chromatin can physically unwind and open up.
Mm-hmm.
But here is the grand finale. Neurog2 then physically acts as a molecular bridge.
A bridge?
Yeah, because of its unique structure, it can bind to the distant enhancer switch, and it can also interact with the machinery at the promoter vault door at the exact same time. It physically grabs both pieces of that stiff DNA wire and pulls them together, forcing the 3D chromatin to loop in the living cell.
That is just stunning. It is the master architect of the cell's fate. It clears the land by stripping the methyl locks, it draws the blueprints by opening the chromatin, and then it physically bends the steel girders to build the new neuron.
It is a remarkable feat of biological engineering.
Let's do our final mini recap to wrap our heads around this last concept.
Sounds good.
Bullet 1. The genome isn't a flat string. It physically loops and bends in 3D space. Bullet 2. It uses insulated rooms called TADs to prevent chaos and keep switches matched to the right doors. And bullet 3, pioneer factors like neurog2 are the heavy machinery, stripping chemical locks and acting as bridges to bend the genome into shape.
That perfectly summarizes the mechanics.
So think about how rearranging the furniture in a room completely changes how people interact in that space. If you move the couch, people walk differently, they talk to different people. The physical 3D structure of your environment dictates behavior.
And the genome is doing the exact same thing to dictate human consciousness.
And that really brings us to the culmination of this massive, elegant study. We have journeyed through the entire sequential birth of a neuron.
From single-cell RNA snapshots,
Right.
watching mathematical waves of gene expression,
To mapping remote dynamic chromatin switches.
proving those switches work in living tissue using electroporation and barcodes.
And finally, seeing how the genome physically strips its chemical locks and folds in 3D space. So, after all of this, what does this all mean? Why should we care so much about dissecting the molecular plumbing of a mid-gestation mouse embryo?
Because this intricate gene regulatory logic is the absolute foundation for understanding how the human brain builds itself.
Right.
When this immensely complex choreography of switches, loops, and pioneer factors goes right, billions of times over in the dark of the womb, you get cognition. You get human consciousness. You get the ability to listen to and comprehend this deep dive.
And conversely, when the timing is just slightly off, when a switch is blocked by a repressor protein, or a 3D loop fails to form because of a mutation, or a protein like LRFN5 isn't expressed at the exact right millisecond, you get the profound neurodevelopmental conditions we see in humans.
Exactly. The roots of autism, muscular dystrophy, or CHARGE syndrome aren't just broken genes. They are disruptions in this beautiful, highly sensitive 3D architectural dance.
Which leaves us with one final, deeply provocative thought to consider, something that stretches far beyond the immediate findings of this specific paper.
Yeah, because we've seen today that pioneer factors like neurog2 possess this almost godlike ability to strip away chemical locks, completely rewire the 3D architecture, and dictate the ultimate fate of a cell.
They are the ultimate biological reprogrammers.
They really are. So what if we could eventually harness that exact mechanism?
Oh, man.
If these specific molecular bridges can force a blank stem cell to build a complex neuron from scratch, could we one day deliver these exact pioneer factors into a damaged adult human brain?
Could we use this genomic rewiring logic to force the adult brain to physically rebuild its own broken circuitry from within?
Could this be the key to finally curing devastating neurodegenerative diseases?
That is the true power of understanding the blueprint. It's not just about satisfying our curiosity about how the factory was originally built. It's about learning the deepest secrets of the machinery so that we might one day be able to repair it. Until next time, keep diving deep.